Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond and become very good at what they do. But only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in fields such as sleep science, cancer, stem cells, ketogenic diets, and more. Get ready. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Carolina Soekmadji. She's a senior research officer working on prostate cancer, the biology of it. She's had a long history in research. She started training in Japan after being accepted into the Panasonic Scholarship Program. And she got her master's degree at Kyoto University on protein structure. Then moved to Australia, where she completed a PhD at the University of Melbourne. And the PhD was about the mechanism of exocytosis and endocytosis of synoptic vesicles. So, Carolina, thanks for coming.
Thank you very much for having me, Richard.
Yeah. Well, tell me about your current research. What's it about?
Yeah, so I'm working on the extracellular vesicles. I think there are a few podcasts already about extracellular vesicles. Is the vesicles that are secreted by the cells. And I'm personally interested into the development of extracellular vesicle-based personalized therapy in prostate cancer and other cancer. So I've been working in prostate cancer for a while, and during my study, we discovered that the extracellular vesicles can actually induce the cell proliferation and survival when prostate cancer cells are under growth without androgens. So maybe if you know a little bit about the androgens in prostate cancer, is that the androgens is
you mean when your testosterone?
Hormone. That's
so
correct.
When testosterone is chronically low.
Exactly. Yeah. So the androgen deprivation therapy, which is actually developed in 1940s, the ideas that came up at 1940s and still is the most preferential strategy for prostate cancer in the clinics, is that when people realize that if we limit the availability of androgens or the testosterone and so on, we can actually inhibit the development or the growth of prostate cancer. So
I thought that, Caroline, a quick question here. I thought that it was chronically low testosterone that promoted prostate cancer. Are you saying this therapy would seek to take away all testosterone and that would stop the cancer?
Well, the androgen deprivation therapy has a various different strategy. Either they inhibit the androgen receptor itself, which is the nuclear receptor that binds to the androgens and drive the gene for proliferations, limit the availability of androgen in the body by inhibiting the secretion of androgens in the body. So that is the original and still the techniques that are being used in the clinic so far. When we starting to look into the extracellular vesicles, and we realized that a subtype of extracellular vesicles can actually increase the proliferation of prostate cancer cells. That's actually one of my main questions personally, is to what extent and how much is the influence of the extracellular vesicles to help the growth of prostate cancer when there are no androgens. And we know that a lot of patients responded quite well with the ADT, but there are some which are not responded very well, and even though there is a reduce in the biomarker for tumor growth, the prostate specific antigen or the PSA.
A question here, I've read that the prostate releases prostasomes, which are like EVs that end up, being coupled with sperm, or they contribute to the seminal fluid. So, is there a diagnostic by which you would get a sperm sample, and based on the condition of the prostasomes, be able to tell the condition of the prostate?
That is also part of it. The prostasomes in what I believe is different vesicles than the small extracellular vesicles that are being secreted by prostate cells. So the prostosomes is known to be involved in the fertility and so on. But what I'm particularly interested in is small extracellular vesicles, which in the past people always mention it as an exosomes. But I'd like to be more cautious about how we call these vesicles, just because even though in the in vitro studies we know that the exosomes is coming from the multivesicular bodies and through the recycling endosomes and being, expelled out. But in terms of tissue or organs, how these vesicles is being secreted by prostate cells and get across into the blood circulation is unclear. So I would rather call it extracellular vesicles at the moment.
Right. It seems like it's unclear the biogenesis of EVs, and also once they enter into a cell, where they go and what happens to them. I mean, I know you studied the exo and endocytosis of at least when the cell membrane buds off and produces them. What did you learn there? Were you studying that?
So there are a few markers that has been used in the past to say that a vesicle is an exosome, which is, CD9, CD63, TSG101, ALEX, and so on. But what was interesting is I noticed that there are differences between these markers that, is not equal among them. So that suggests that there are actually various or multiple groups of small extracellular vesicles. And that makes the study is much more complicated, because if we want to look into the role of extracellular vesicles, I think it's really important to know which vesicles we are looking at.
So again, from what I know, some vesicles come because the cell membrane buds off, and then some come from the interior of the cell. So what is your research hoping to figure out about EVs in particular? Again, the biogenesis of them in prostate cells, or the fate of them once they enter in?
Yeah, so interestingly, if we grow the cells in the presence and absence of androgens, and the one that are different is the CD9 instead of the other vesicles. So it looks like these CD9 enriched vesicles is the one that are secreted more when the cells are treated with the androgens. And when we put back the CD9 enriched vesicles into cells that grows without the androgen, we also observe an increase of proliferation. That suggests that even in the small extracellular vesicles group, the CD9 positive enriched vesicles is the one that are likely to be responsible for the response of cells to keep growing even without the androgens. So that's bring a sort of informative point about why certain patients likely to respond to the ADT and why some didn't. Why they can still have prostate cancer that are grown even though there is no androgen. Would the extracellular vesicles actually play a big role apart from the other things that we already know, like gene mutations, and all sorts?
What's weird though is, throughout a man's life cycle, he's going to have very high androgens in his teens and 20s, and then less in his 30s and 40s, and so it appears that, I guess, prostate cancer appears to correlate with persistent low levels of androgens. So why wouldn't it happen earlier if androgens kind of correlate with the spread of the cancer? It seems like a new regime has come into place where, the prostate's functioning in a different way now. And that's why the presence of androgens make the cancer grow. It's odd, it seems counterintuitive.
Yeah, that is actually a good point, yeah. So for some men who has a history of prostate cancer, they do develop or likely to develop prostate cancer much earlier because they harbor that mutation that are being passed from, their parents. But it is known that there's a lot of other factors where, the cells itself growing old. So then the cell cycle regulation, DNA replication, and so on, starting to get into a lot of problems. Or also the influence of lifestyles is also being discussed. But it seems like at some point, men will develop some issue in their prostate because of their because they're just getting old. It's just the way the life is. Ideally, I guess the if man wants to go through prostatectomy, they wouldn't need to worry about prostate cancer. But we know that prostatectomies also have possibly side effects that people or patients need to consider whether that is the right treatment options for them or not.
Oh, what are the side effects of taking out your prostate?
Similar to other risks for surgery, and additionally, there are some cases reported where patient might experience erectile dysfunction or uncontrollably passing urine, and that needs some sort of therapy to get them back into the normal physiology. So that's sort of risk which I don't think all men consider it very lightly.
Okay, well, back to the androgen therapy. Of the men that have prostate cancer, do they tend to have low testosterone at the same time, or is there no correlation between their circulating testosterone levels and the presence of prostate cancer?
Well, there's some looking into the circulating testosterones, but what we are looking at is actually the correlations, how the prostate-specific antigen, which is being secreted by cells as a marker for tumor growth. So, probably I didn't really explain that very well. I guess it's not really what I'm researching right now. Yeah, and
I just wonder if there was that. I just wonder if there was that correlation. But like you said, in the absence of pretty much all androgen, what happens to the cancer with the therapy? And when it works in people, does it completely go away, or does it just go dormant? Like, what happens?
Well, depending on everybody is different. So there's quite a lot of cases where people responded very well, so the prostate cancer just go away after first treatment. But there are a few that eventually has an increased prostate-specific antigen or PSA, and that suggests that there is a growth of prostate tumor, and that's also held true in the advanced prostate cancer, where prostate cancer is actually not in the prostate anymore, but in different sites of the body that they still have a high PSA in their blood.
Okay, so what is it about the EVs specifically that you're looking to figure out, in the case of prostate cancer?
Yeah, so so for the extracellular vesicles, we are looking into how these EVs is actually playing a role in the proliferation and survival of prostate cancer. So, by inhibiting of the androgens, we know that the prostate cancer gets, they could either go to cell death or they're going into cell senescence. However, there are some which are grow back. So we are looking into the role of the extracellular vesicles in this process, whether they are playing a role in these prostate cancer cells to grow back despite the absence of androgens.
Well, where does prostate cancer tend to metastasize when it does?
Yeah, so the prostate cancer tends to metastasize to bone, and they also can be found in the advanced cancer in other disease, in other organs like the liver or the lung, but the majority is in the bone. So we are looking into how the bone environment or the extracellular vesicles derived from cells that are grown in the bone can influence the growth of prostate cancer as well.
Yeah, it seems like that the EVs, let's say from prostate cancer will circulate, and then they'll enter into the target cells, bone,
that's true. Yeah.
whatever it may be, and then change the expression of the bone cells so that now when there's circulating cancer cells, they'll be attracted to that area and take up residence there and form a metastasis. So it seems like the exosomes are doing niche construction, early niche construction.
Yeah, that is true. Yeah, there's some preclinical mouse model that shows that indeed is what happened, that when extracellular vesicles form, cancer cells is being introduced into the circulations, they seem to harbor into certain sites, and depending on the integrin profiles, they can go to specific type of tissue or organs. In our study itself, we do look into the integrin profiles, but we also look into other sort of molecules that potentially play a role in that process. Perhaps there are some other things that are important.
Okay. What do you feel like you're close to figuring out in regards to the EVs? What do you understand about them better than anyone else so far? Do you feel like you're getting close to an understanding or a breakthrough in the next few months or a year? Or is it going to be a long time, you think, before there's more understanding of what's going on?
Well, there are a few things that are quite interesting to follow it up, apart from the potentially integrins involvement, but also what subtype of vesicles that are playing a role in this process, and whether they are the same vesicles that are involved in the proliferation, but also are they also involved in the metastasis process, and probably a different type of vesicles. And the other thing that I'm looking at is actually how to match the EV profile with the treatment that are being given to the patients, because we know that some patient doesn't respond and some patient responded. So we'd like to know whether we can stratify this patient based on the extracellular vesicle profile in the blood samples.
Well, I've heard that most tumors are very heterogeneous. So if you fully understood all the clonal lineages of a given tumor, I wonder how the EVs differ amongst the different mutated cells or amongst the different clonal lineages. I would think that would make a huge difference depending on what the tumor looks like, and how heterogeneous, etc.
Yeah, that is correct. So we are looking into differentiate those, based on the stage of the prostate cancer. That's what we can do, that we can look into the EV profile. But I know that a lot of technology now trying to look into single extracellular vesicles, which we are getting there, but not quite there yet. And it really depending on the development of the technologies that we can all be able to understand this, because they are very small, and our available technology to analyze protein, RNA, and lipid, they have limits, which currently is hard to achieve by analyzing a small amount of material that are available, in a single extracellular vesicles. So at the current stage, what we can do is looking into the type of fingerprint type, so it's sort of like a barcode coming from the profile of the proteins, the RNA, and the lipid, potentially, whether we can do that and help to stratify this prostate cancer and how individually people may respond to that.
Very interesting. So what's the best way for people to learn more about your research and to get in touch with you?
Well, I have my profile available on Twitter in a link, and also in the Institute website. So can send me email anytime. We are very interested to look into a treatment response in the 3D models, and also looking into these molecules that are coming out from the blood samples. So anyone who's interested to learn more about that, we can certainly have a chat.
Okay, well, very good. Well, Carolina, thank you for coming on the podcast. I appreciate it.
Yeah, thanks very much, Richard, for having me.
Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius. 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond and become very good at what they do. But only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in fields such as sleep science, cancer, stem cells, ketogenic diets, and more. Get ready. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.